Hi, this is Alex from devstories.eu. Scanpy Single Cell Analysis, episode 13 of 16. UMAP is fantastic for separating distinct cell types, but it can violently tear apart continuous biological processes. When you are studying cellular development, you do not want disjointed islands of cells. You need to see the smooth continuum of state changes. Exploring manifolds with diffusion maps resolves this exact problem. In Scanpy, you calculate this using the tool diffmap. Users often default to UMAP for all dimensionality reduction. Understand that UMAP is an embedding technique optimized for finding distinct clusters and preserving local neighborhoods. Diffusion maps are fundamentally different. They preserve the continuous mathematical probability of transitioning between states. This makes them the ideal choice for analyzing continuous processes like cellular differentiation. The diffmap algorithm treats your manifold as a continuous network. It relies entirely on the nearest neighbor graph of your cells. Once that graph is established, the algorithm simulates a random walk across the connections. Think of it as modeling a diffusion process, similar to how heat spreads through a physical material. The algorithm evaluates how easily a signal can travel through the dense regions of your data. It calculates the probability of moving from one cell state to another over a specific number of steps. Cells that share a high probability of transition are placed closer together in the final lower dimensional space. Consider tracing a hematopoietic stem cell differentiating into an erythrocyte. If you project this data using an embedding that favors discrete separation, the intermediate progenitor cells often get forced into artificial separate clusters. The underlying math fractures the biological timeline. If you run diffmap instead, the algorithm computes the transition probabilities along the entire developmental path. The result is a smooth, continuous trajectory. The stem cell sits at one end, the mature erythrocyte at the other. Every intermediate state is plotted along a connected path based strictly on the likelihood of state transition. You are not looking at isolated snapshots of distinct cell types. You are looking at a fluid biological event. Applying this in Scanpy follows a rigid sequence. First, you must compute the neighborhood graph in your object. The diffusion map cannot run without those pre-computed neighbor connections. Next, you call the diffmap tool and pass it your ANData object. You can optionally specify the number of components you want to compute, which sets the dimensions of the output. Scanpy calculates the diffusion map and stores the new coordinates in the multi-dimensional observation attribute of your object under the key xDiffMap. The tool also stores the eigenvalues in the unstructured data attribute. These values tell you how much variance each diffusion component captures. A sharp drop in these eigenvalues indicates that you have captured the most important biological transitions, and the subsequent components are likely noise. In a typical differentiation dataset, the first non-trivial diffusion component directly aligns with the primary developmental time axis. Here is the key insight. In a diffusion map, the physical distance between two cells on the plot is not just a generic measure of transcriptomic similarity. That distance explicitly represents the mathematical probability of a biological transition occurring between those two specific states. That is your lot for this one. Catch you next time.